Are you okay? Sorry, sir. I'm very sorry for your loss, sir. I wish we could detect the cancer cells earlier, but I'm very sorry, sir. We have done our best.
I think we can.
What?
There is something called
APTAMERS.
So aptamers, what are actually aptamers? Aptamers are short, single-stranded DNA or RNA that can bind selectively to specific targets such as peptides, carbohydrates and also toxins. Aptamers and antibodies, they are quite similar to each other. For the past decade and for the past few centuries, antibodies have been used for the treatment and diagnosis of cancer. However, aptamers came into the equation. They have more advantages as compared to antibodies. They are more specific, alright, and at the same time it has low immunogenicity. Therefore, they work more well for cancer. So, there are a few applications of aptamers, which include firstly, the formation of new drug, also involvement in the drug delivery system, including as well for diagnosis of disease. And then the fourth one, it was also used in bioimaging, analytical reagent, and lastly is also included in food inspection. Wow, so aptamers are quite interesting. So you guys want to know more about it? I have a place where we can get more info about aptamers. Let's go to a lab located in Philadelphia. Let's go!
Okay, ladies and gentlemen, I'm reporting live from Philadelphia State in USA. So I'm actually in a lab located in this particular state, which is believed to be one of the places where aptamers were found. Cancer is the second leading cause of death around the world. Therefore, researchers have been working day and night to investigate improved therapeutics as well as early detection methods for cancer. Therefore, aptamers are believed to be the solution to this problem. You want to know more about aptamers? Let's find out. Ladies and gentlemen, so now I'm going to interview a man, a very important man, which is also a research pharmacist, which will actually explain to us how aptamers are actually manufactured. So let's come. Hello, Mr. Idlan. Your name is Mr. Idlan, right? So the audience and me, we would like to know how actually these aptamers, so-called aptamers, the solutions, are actually manufactured.
Thank you, Ubin. Okay, so APTAMERS is actually being produced by a system known as SELEX. So what is SELEX? SELEX is an acronym of systematic evolution of ligands by exponential enrichment. Okay, so basically it is consists of 5 steps, which are preparation, incubation, partitioning or elution, and the fourth one will be the amplification, and lastly will be the sequencing. Okay, so for the preparation, okay, this is the preparation which consists of random pool of about 1,000 of DNA sequences, and next will be the incubation. Okay, so incubation means that it is a folding of primary to secondary or tertiary, or we known as from 2D structure to 3D structure. And in these steps also, there will be a formation of complex by binding of the aptamers and also the target, which will produce bound sequences. And next will be the partitioning, and then before that, the unbound sequences will be removed or washed away, and the bounded sequences will be next amplified during the amplification step, and the products of amplification will be used in a sequencing steps. Okay, in these sequencing steps, it is used for the next selection cycle, and the same cycle will be done up to 15 times. I think that's all for the steps of manufacturing aptamers. Okay.
Alright, thank you so much, Mr. Idlan. So I hope now it's clear how these aptamers are actually manufactured. So now we have taken a look at how they are manufactured. It's time to take a look at what is actually the mechanism of action of aptamers. How do they actually work? Let's find out. So one of the application of APTAMERS is diagnosis of cancer cells. So now we want to know how cancer cells can be diagnosed by using APTAMERS. So now we are going to interview right over here another very important research pharmacist, Mr. Fadlizil.
Oh, hi.
Hi. Nice to see you, sir.
Yeah, my name is Fadli.
Okay.
Researcher for the APTAMERS.
Okay.
Okay.
So this is a schematic illustration of microfluidic device showing a middle region with immobilized aptamers. And there is a label of cancer cell, normal cell, aptamers, and microfluidic device. So after the injection of the sample, cancer cells are trapped by specific aptamer cell interaction, whereas normal cell pass through the device. In this sense, cancer cell can be detected by optical microscope observation of the middle zone, and also elute of cancer cell can be used for other studies. So how can we see the cancer cells? The first step is the aptamers will be labeled with the fluorophore to become a fluorophore labeled aptamers. And then this molecule could be used for fluorescent staining of cancer cell as the top image, in vitro cancer cell imaging, and as well as in vivo cancer cell imaging in the mice, like this. And we can see the imaging, the result of the diagnosis is like this.
Alright, so now it's very clear how important aptamers can be when it comes to cancer. We all understand that really well now. Thank you so much, Mr. Fadlizil, for explaining us.
Okay, thank
Alright,
you.
thank you
Mr.
so much.
Mubin.
I appreciate your time, okay?
Okay, thank you.
Alright, thank you.